do that again. Okay, so I haven't said anything very important so far, except that membranes enclose cells and they enclose organelles, and by controlling the flow of molecules or acting as a barrier to the flow of molecules, they can concentrate things within compartments, either the cell or an organelle within the cell, and they can play many roles in cell signaling, and so we'll talk about a little bit of that today. This is a very kind of like the most simplified view of a membrane as just a barrier that enables cells to concentrate molecules or exclude molecules, and organelles within the cell have the same ability to concentrate or exclude specific components, and by doing that, cells can compartmentalize their chemistry, which is enables them to do a lot of things that would be tricky if everything were happening in the same compartment and just controlled by diffusion. So today we're going to talk about the structure of the membrane and a little bit about membrane proteins. And then in a lecture next week, we'll talk about the role of especially proteins within the membrane in cell signaling. I'm not going to talk about transport very much, but which is, I think, chapter 12 in the book, but it is an important topic. I will talk about protein trafficking, which is how proteins get to specific organelles within the cell and go inside and out of the cell. Of course, cells are very plastic. They're moving often and they're growing and shrinking. So it's sort of evident that the membrane has to be a very plastic or dynamic structure that can easily grow in surface area and change in shape through, and there's zillions of proteins within the cell whose role is to shape membranes and control the budding and fusion of membranes to control the movement of membrane associated and internal proteins around the cell. So we'll come to that next week. So today we're really focusing more, as I said, on the structure of the membrane. And the membrane is this kind of amazing substance, which is a bilayer. The bilayer of earthly cell membranes is composed of lipid molecules. So there's kind of this mirror image structure to the membrane where there's two halves, and the topology is very important. And then these just represent proteins that are stuck in the membranes, and we'll talk more about how membranes, how proteins and membranes interact later. Okay, so membranes are very thin. 5 nanometers is extremely thin. That's sort of, that's the diameter of a small protein would be about 5 nanometers. So some proteins can span the membrane and have domains on both sides. So this is actually from chapter 2 in the book, which I didn't have you read, which is more about the building blocks of life, which hopefully you learned in other courses, but this is just to kind of remind you that these are the main macromolecules in cells, and we've talked a little bit about nucleic acids and proteins. We'll talk very glancingly about polysaccharides today, but mostly we're focusing on lipids, which are built from fatty acids. And fatty acids are molecules that look like this. They have long hydrocarbon chains, which are hydrophobic. They do not like water. And then they have a reactive group that enables them to be attached to different things. And the main thing that they're attached to with respect to the membrane is glycerol, which is a 3-carbon triol. So it's 3 carbons with alcohol, with OH groups on each carbon. And so glycerol can be modified by the addition of up to 3 fatty acids. When there are 3 fatty acids attached to a glycerol kind of core, that is triacylglycerol. And that is a hydrophobic molecule. It's an oil or a fat. And within cells, triacylglycerols form little droplets, and they are kind of the storage droplets for the building blocks of lipids, of membrane lipids. And so when a triacylglycerol is converted to a phospholipid, one of the fatty acids is cleaved off, and the free hydroxyl group is then modified by other groups that are water-loving rather than water-hating. And so there are a few examples in the textbook, and I'll show you a couple of those. But this amphipathic nature of the the lipid molecules is essential for the structure and function of membranes. The fact that this side likes to be near water and this side hates to be near water basically dictates everything about membranes. So this is the most common lipid in cell membranes, at least in animal cells, where the third hydroxyl group on glycerol has a phosphate group and then a choline nitrogen-containing group. And so this together forms the hydrophilic or polar head group that faces outward within the membrane. And this forms the hydrophobic group that faces inward away from the aqueous environment of the interior of the cell or the exterior of the cell. So the fact that life is built in an aqueous environment is what enables membranes to do their job. This part likes the water, this part hates the water. Okay, so membranes are heterogeneous. Different membranes in the cell contain different compositions of phospholipids. And so this is phosphatidylserine. I showed you phosphatidylcholine in the previous slide. Different mixes of lipids give different membranes within the cell their character. And in addition to these phospholipids, there are additional components within membranes that include, at least in animal cells, sterols. So this is cholesterol, which is a very well-known and very important component of animal membranes. And this is a galacto or a glycolipid. So it's also got these hydrocarbon tails, but instead of the phosphocholine or phosphatidylserine head group, it's got like a sugar and some other stuff here going on. So these are all things that are incorporated into membranes, and they all have this amphipathic property where they like water on one side and hate it on the other. And that gives them a defined orientation within the membrane, because the water-loving side always has to be at the exterior where the water is. And this part basically excludes water by packing together so that the water is excluded from the volume of the membrane. So in plant membranes, most of the phospholipids contain at least one hydrocarbon tail that has one or more double bonds. And that's called unsaturated. Right? So you can add hydrogen to a double bond, and that saturates it. That's called a saturation reaction or a hydrogenation reaction. In naturally occurring membranes, most of these double bonds are in the cis configuration, so they kink the membrane like this. Actually, when I was an undergrad at Berkeley, I had this slightly crazy professor who taught me, who taught the lab class that I took, and he told us something that was so far ahead of his time, which was that so often people back in the Depression and thereafter, they when butter was hard to come by, they started hydrogenating vegetable oils to get rid of some of these double bonds. And what wound up happening is that the chemistry of that hydrogenation process would create aberrant kinds of structures that don't naturally occur in nature, which we now call trans fats. And he told us we should not eat trans fats because they were not natural and they were bad for us. And nobody at the time was talking about trans fats, but now we talk about trans fats. Anyway, so it's these molecules that can be in a cis or trans conformation, and the chemistry of hydrogenation in which a more liquid, a more unsaturated, kinked fatty acid is converted to a more saturated fatty acid can also wind up esterifying or changing isomerizing these double bonds and turning them into the trans configuration. Okay, so someone is not muted, so I'm gonna make sure everyone's muted. I can't hear you, but I guess someone's hearing this. Okay, so these double bonds, these unsaturated fatty acids have a naturally more kinked structure, and that actually limits, now I can't get my mouse onto my screen. Okay, that limits how tightly these lipids can pack within the membrane layer and makes them more fluid. It increases their mobility. And so the degree of saturation or unsaturation is tightly controlled by different cells to control the fluidity of the membrane. And organisms that have evolved to live at higher temperature, like animals compared to plants, for example, tend to have more saturated hydrocarbon tails because at higher temperature, you actually need to saturate more of the bonds to have the right kind of fluidity. So plants don't have, so the degree of saturation varies in different cell types. And another way that the composition and the fluidity of membranes is controlled is by adding additional components, particularly cholesterol, which I mentioned earlier, this sterol. And this kind of counterbalances the kinks that are created in the tails by these double bonds. The cholesterol molecule can kind of pack into the gaps that are created by the kinked tails and sort of fill them up and make the membrane more stiff and thereby less fluid. And it also cholesterols also change the permeability of the membrane to other molecules. So animals have cholesterol in their membranes, plants do not have it. I always kind of think it's weird when you go into a market and you see this cholesterol-free popcorn, but of course, popcorn, unless you add butter to it, does not have cholesterol because it's a plant. Okay, so I already sort of mentioned this, that the building blocks for these phospholipids are triacylglycerols, which are stored as little oily droplets in cytoplasm or in fat storage organs. And this is just another view of the membrane showing the lipid bilayer. It's just showing water on the two surfaces to highlight the fact that the hydrophilic phosphate head groups are in contact with water, and the hydrophobic tails are inside, and the water is excluded from the interior of the membrane. So this is a fluid substance. It's sometimes called a liquid crystal because it has the molecules within it can easily move, but the lipid molecules tend to only move within the layer that they're in. They almost never spontaneously flip from one side to the other because energetically that would require the movement of one of these hydrophilic groups to the other side, which is energetically extremely costly. So it does not happen spontaneously. And that's just what this shows. And so the membrane, as I said, has to be very adaptable.
To demonstrate the fluidity of the lipid bilayer,
I'm gonna let you listen to this.
A piece of the plasma membrane of this neuronal cell is pulled out with laser tweezers. Remarkably, moving this membrane tubule rapidly back and forth does not rupture the plasma membrane, which flows quickly to adapt to the mechanical distortion.
So pretty cool, right? So you can visualize membrane dynamics in a variety of ways. That fluidity is extremely important for everything membranes do. The fact that this interior part of, the interior part of the membrane hates to be exposed to water also controls the shape of different vesicles, that the membranes will tend to self-seal very, very rapidly so if they get ripped, they'll immediately seal up and form a new compartment that will completely separate the hydrophobic hydrocarbon tails from any aqueous environment. It's just energetically very, very costly to have those hydrophobic tails in contact with water, and that's called the hydrophobic effect. Okay, so membranes are synthesized. The chapter really does not touch on the synthesis of these phospholipids, and I'm not going to, I don't know much about it myself, but it just mentions, this is like taken verbatim from the figure legend, that there are enzymes that are bound to the cytosolic monolayer of the ER membrane. So we haven't really talked about the ER yet, but hopefully you remember that that's this like, the bulk of the membrane inside at least an animal cell is the endoplasmic reticulum, this crazy reticulated or net-like network of membranes. And on the cytoplasmic, so compartments within the cell have typically one side faces the cytoplasm and the other side is the interior of the compartment, and that's called the lumen. So the proteins that make these phospholipids are on the cytosolic or cytoplasmic side of the ER membrane. They synthesize phospholipids from free fatty acids that are derived from triacylglycerols, and they insert them into the monolayer. And then there are scramblases that basically scramble the different kinds of flavors of phospholipids and sort of even out the composition of the membrane. And then there are sort of the reciprocal kind of enzyme that can vary in a very orderly way, can in a biased way, they can move specific phospholipids from one side or leaflet of the membrane to the other side, whoopsie, and thereby control the composition of the two halves of the membrane, which are often very asymmetrical. Okay, I think that's all I wanted to say about that. So probably worth knowing about scramblases and flipases, although the actual mechanism of how they work is beyond the scope of this course. Okay, so for the rest of this, I'm going to talk, I guess, mostly about membrane proteins, and we'll have more to say about membrane proteins when I talk about signaling. Proteins, which are just shown in different flavors here, can associate with membranes in a wide variety of ways. They can span the membrane just once and have just a single helix that passes through the membrane and have stuff on both sides. They can have much more extensive contacts with the membrane that typically have multiple helices embedded within the membrane to form a pore or a channel. And they can also be associated more peripherally with the membrane. And I'll show you at least a few ways that can happen in a few slides. So when proteins span the membrane, the part that spans the membrane is typically organized as a single alpha helix or a set of alpha helices. And because these alpha helices are in direct contact with the hydrophobic tails of the lipid molecules, the amino acids that face outward from this helix that are contacting the lipids tend to be hydrophobic amino acids. And so it's actually quite easy to, I'm not sure if I put in this video, I think I did. Computationally, if you take a protein sequence and run it through a transmembrane prediction program, it can tell you where there are likely to be these transmembrane helices. So, and they're typically about, I think the median is 24 amino acids long, and they range from actually a little bit bigger than this, probably 20 to 27 amino acids or something like that. So, because that's how many amino acids you need to make a coil that spans this 5 nanometer distance through the membrane. Okay, and sometimes there are other structures, which I'm not going to talk about. I'll come back and talk more about this in a future slide. Some proteins don't have intrinsic, don't have regions that intrinsically associate with membranes, but they get modified with lipophilic groups that do like to stick to the membrane, and so they get peripherally anchored to the membrane through those non-amino acid groups. Okay, so this is just a table from the chapter of different kinds of important proteins that reside within the membrane. I think these are all plasma membrane proteins, but there are zillions of proteins that are in not just the plasma membrane, but the membranes of all the different organelles in the cell. And we'll talk about how they get there, as I said, next week. Okay, so this just kind of reiterates what I just said, that this just shows a sort of higher resolution, an alpha helical segment of a protein that is projecting through the lipid bilayer. And again, these amino acids, that the side chains on those amino acids that are in contact with the hydrophobic groups of the lipids have to be hydrophobic, that the backbone of the peptide is relatively hydrophilic. And so this really has to be these have to be hydrophobic to like being in contact with this non-aqueous lipid environment. There are also cases where, as I said, where you can have multiple helices that span the membrane, and this is threaded through multiple times. So this would be like a five pass transmembrane protein. And if the function of the protein is to form a channel, often it'll look like this, where each helix will have a hydrophobic face that faces outward toward the hydrophobic lipid groups, but the interior may be a pore that is actually hydrophilic, that allows water to flow, or other hydrophilic compounds like proteins. Actually more like ions. Passing a whole protein is something that is relatively rare, and that's more like the function of the nuclear pore. So scratch what I just said. Okay, so you can recognize, again, the computational prediction programs can identify helices that are either hydrophobic, like this one, or amphipathic, again, the same word, amphipathic, meaning it both likes and hates water. The side likes it, the side hates it.
I made this short video clip just to illustrate how a computer program finds transmembrane helices. Let's say I'm reading a paper about a C. elegans protein called fluid 1, and I want to see the transmembrane domains in this protein. So I need the amino acid sequence, which I get from Wormbase. This is the repository for all the C. elegans genome and genetic information. And I go to fluid 1 and I click to look at the amino acid sequence, and I just copy and paste that into a web browser for a program called TMHMM, Transmembrane Hidden Markov Model. And very quickly it will just analyze the protein sequence and show me with these red bars the likely locations of transmembrane helices.
Part of the chapter talks about the challenge of studying the structure of membrane proteins. And this was one of the first membrane proteins that really was, our peripheral membrane proteins, also alpha helices. Alpha helices are really, really common that's one of the most common structural motifs in proteins. So, but peripheral membrane proteins can have any structure, basically, and they can have both alpha helices, beta sheets, and whatever other kinds of structures that proteins have. It's just that the little rods that pass through membranes tend to be alpha helices.
Abby, someone else has another question too. Can cholesterol be added to mostly saturated membrane to make it more fluid?
No, actually, cholesterol actually has the opposite effect. It makes it less fluid. It kind of packs the membrane more tightly so that it moves less. And I don't really know very much about how cells balance the amount of cholesterol and unsaturated bonds in their membranes. I just know that both contribute to modulating the fluidity of the membrane. Okay. So this is a super cool membrane protein that is also the ancestor, or I shouldn't say the ancestor, but it is a homologue of the proteins in our eyes that enable us to see light. So it's a protein that sits in the plasma membrane of a class of halobacteria, which are not actually bacteria; they're actually archaea. They used to be thought to be bacteria, but are now recognized as archaea. And this bacteria rhodopsin has a light sensitive it undergoes a light sensitive conformational change that allows protons to pass through this channel. And by controlling the flow of protons, this protein can turn light energy directly into a chemical energy by creating a gradient of protons, which it can then harness by allowing the protons to pass back through the membrane. It can use that stored chemical energy to do work. And you've probably seen these organisms and maybe not realized it because they concentrate in these salt flats that are at the southern end of the San Francisco Bay, where there used to be, actually there still are salt mining operations, because these are concentrating ponds for salt. They're extremely saline, and that's where these organisms like to live. And these organisms give these pools their reddish or purplish color. And they have this retinal, which is a chromophore that is what actually absorbs the light and undergoes a conformational change. It's very similar to the chemistry of the rhodopsin in our eyes that also undergoes this light-induced conformational change to generate a signal which we see or we interpret as light. Okay, so I mentioned that proteins can have sort of intrinsic hydrophobic helices that wind up getting them inserted into membranes. But some proteins also get attached to membranes by being modified by the addition of chemical groups that are hydrophobic and like to be stuck into the hydrophobic environment of the membrane. And these are a few examples. GPI, which I'm blanking on exactly what that stands for. I knew like 5 minutes before lecture started, but this some proteins are GPI anchored. Some proteins are modified by groups that are what are collectively called prenyl groups. They're made of a repeating subunit called a prenyl group, and those are farnesyl or geranyl groups. And then these are other kinds of chemical modifications, palmitoyl and myristoyl, that can be added to different sites on proteins that then attach them to membranes. And they are attached by different kinds of enzymatic pathways to different parts of the protein. Like this one shows attachment to the N-terminus of the protein, the very N-terminus. This group is attached to serine residues within, like anywhere within the sequence of the protein. And these groups are generally attached to a particular, to the C-terminus of the protein, which has to have a specific CAAX motif, or I'll show you what that looks like in just a second. And I'm mentioning this is actually not in the chapter, so this is a little kind of addition. But I'm mentioning this partly because I talked last lecture about the nuclear lamina, and I showed this micrograph of these filaments that form this kind of cross-linked meshwork that is on the interior of the nuclear membrane. And it turns out that that as this is assembling, so after cell division, or during cell division, this gets taken apart and then it gets reassembled as the cell kind of goes back into interphase. And the lamins get associated with the inner nuclear membrane by being farnesylated. This is, again, one of these prenyl polymers that are synthesized and then added to proteins that have this particular motif, CXXX basically, motif at their very end. So lamin A, which is, there's more than one lamin, but I won't deal with that right now. Lamin A has to be farnesylated by a farnesyl transferase to be efficiently attached to the nuclear membrane. And then this lamin protein actually is only farnesylated transiently once it's associated with the membrane and forms this this cross-linked basket-like structure, the lamina. This farnesyl group is actually removed, and the way it's removed is that there's a cleavage site for a protease that's upstream of this attachment point, so that the very end of the protein, the C-terminus of the protein, along with the farnesyl group, gets cleaved off. And what does the AA mean in CAAX? I think it means any amino acid. And I actually should have looked this up. I don't remember what the CAAX designation is, so I will look that up.
I thought that the X meant any amino acid.
Usually it does, but then what is the AA?
Yeah, I'm trying. I'm like looking it up right now.
Okay, thank you. Okay, so
I
okay.
only see. Yeah. Anyways, we'll find it out later.
Yeah. Okay, so I'm mentioning this partly because of this really remarkable, I mentioned that mutations in lamin proteins have been associated with a variety of different diseases. And it turns out that certain kinds of mutations that mess up that farnesylation pathway can give rise to a really unique premature aging syndrome called Hutchinson-Gilford progeria syndrome, or HGPS. In which basically kids who, they start to age prematurely, they develop most of the hallmarks of aging, their skin gets wrinkly, they lose their subcutaneous fat, and they die very young of symptoms that are associated with age, heart attacks, strokes, atherosclerosis. So it's a really strange syndrome, and it was really kind of striking when it was identified at the molecular level and shown to be due to mutations in the lamin A gene. And it turns out that I showed you this pathway for the modification of the lamin A protein. It turns out that the mutations that cause this HGPS typically they either, usually through a splicing mutation, they mess up the cleavage sequence that normally leads to this being removed. And we actually do not yet understand why these patients age prematurely, but their nuclei, instead of having a nice smooth nuclear envelope, wind up having these really, especially in certain tissues, they tend to have very corrugated looking nuclear envelopes, but it's not clear exactly why that leads to this aging syndrome. Thank you, Kirsten. She's on the chat, she answered the CAAX mystery. So I wanted to talk briefly about this, this technique that's mentioned in the chapter called FRAP, fluorescence recovery after photobleaching, because I think it's useful to understand it. And it's a powerful method, not just for looking at membranes, but it's particularly used in the field of membrane biology, because it's really good at measuring dynamics of things especially in a 2D kind of environment like a membrane. So if you want to measure how mobile a protein is within a membrane, you can also look at lipids this way, but for the time being, we're talking about proteins. A common technique is to label these proteins by fusing them to green fluorescent protein or another fluorescent protein. And I'll explain what that is in a second in case this is not something you've encountered before. And then I'll come back and explain this slide. So I explained how fluorescence works a little bit, and hopefully this is not the very first time that you've ever encountered fluorescence, but you probably know, just because you've seen zillions of fluorescence micrographs, that fluorescence requires special dyes that fluoresce, and these dyes can be attached to different structures, as I told you, in cells, which enables you to see things like DNA or microtubules. And the way fluorescence works is that, so it's an intrinsic property of some dye molecules. So a normal, like a dye that just is a color, will absorb photons and will thus look like all the colors that it's not absorbing. But a fluorescent molecule will absorb photons and then undergo this internal conformational change that's called non-radiative decay, that actually releases some energy, but not in the form of light. And then it will emit a photon of light that, because some of the energy has been lost, the photon that is emitted is lower energy than the photon that was absorbed, and lower energy means longer wavelength. And so fluorescent molecules typically absorb light over a range of wavelengths and then emit light of longer wavelengths. And so a lot of the molecules that we use in biology research are things like fluorescein or rhodamine. Those are dyes that have particularly nice abilities to do this, to absorb light of specific wavelengths and emit light of other wavelengths. So to understand FRAP, you also have to know that when a fluorescent molecule absorbs a photon, usually what you want it to do is to do what I said, to undergo this non-radiative decay and emit a photon. But every time it gets excited, you've excited the molecule to a new state, it can go through a different pathway. It can react with oxygen and undergo like an irreversible chemical change that makes it non-fluorescent. And that's called photobleaching. And it's important to understand that that is permanent. It's not blinking of the fluorescence, it's a permanent change so that that fluorophore is never going to be alive again. Okay, and so one of the fluorophores that we use a lot now in biology is green fluorescent protein. So this is different from the small molecule fluorophores that were used up till like the 90s and are still used quite a lot, like fluorescein and rhodamine. This is a protein that was discovered in a jellyfish, Aquaria victoria, which lives all over the world, but I think especially around Victoria, Canada. So it doesn't quite look like this, but it doesn't actually fluoresce. I mean, so it fluoresces, but it doesn't quite look like this. So this protein was it was recognized that this jellyfish contained an intrinsically fluorescent protein, and it was the sequence of the protein was eventually determined, and it was remarkably found that you could take that sequence from the jellyfish protein and put it into a wide variety of other cells, and it would, that would lead to the synthesis of this protein, which underwent this maturation process to become fluorescent. And you can put it in anything. You can put it in any organism or any cells you want to study. You can target it anywhere, and it's been used to do all kinds of crazy things, make fluorescent fish and pigs. And it's been used in every experimental system that people study. And what's really nice about it, so the dyes that we historically use for fluorescence are small molecules, but they're kind of hard to get attached to things inside cells. Whereas this is a protein, and so you can actually create a gene that fuses a protein that you want to study to this protein, and then your protein that you're interested in is now tagged with a green fluorescent signal. And also this protein, so the fluorophore is actually in the center. It's this little thing right here, but this barrel shape, it's literally called a beta barrel, that surrounds it, protects it from the cellular environment and reduces its photobleaching. So it's a really nice fluorophore. Osamu Shimomura was the person who first identified this protein in the jellyfish. Roger Chen, who actually started his faculty position at Berkeley many years ago and then moved to UC San Diego, did a huge amount of work to engineer green fluorescent protein and make it more versatile. He made all kinds of variants that were sensitive to calcium or pH, and also changed the wavelength of it through protein engineering. And Marty Chalfie actually, all he did really was he took this protein and put it into C. elegans and showed that it worked there. But he shared the prize that was given for the discovery and development of GFP as a tool for biological research. And this just highlights that fluorescent molecules tend to have, that they're different ones have different structures, but when you look at them, you'll notice some similarities, which is that they generally have these networks of double bonds, so extended double bond networks, conjugated double bonds. And that enables the electrons to adopt this kind of cloud-like configuration, which promotes this kind of non-radiative decay and this fluorescent property. So these are fluorescein and rhodamine, which are two of the most commonly used sort of inorganic or non-natural synthetic dyes that we use for fluorescence microscopy. And this just points out that, are you guys seeing this control panel? I can't move it off the screen, unfortunately. That the chromophore, the part that actually makes the little thing inside GFP that actually makes it fluorescent, actually looks chemically quite a bit like these molecules, but this is made of amino acids. It's made of these 3 amino acids, tyrosine, glycine, and serine, that undergo this called a cyclization reaction because it creates this kind of cyclic structure, and that's what gives GFP its fluorescence. And there it is, kind of stuck in the middle of the GFP molecule. Okay, that's what you need to understand FRAP, is that GFP is a fluorescent protein. You can stick it onto a membrane protein that you're interested in, and then you can use this technique where you photo bleach a small patch on the membrane using an intense light. So basically you kill the fluorescence of these GFPs, and then you basically look for the fluorescence to recover in that patch. And it's recovering not because these are turning back on, they're dead, they're never going to turn back on, but they're exchanging with the ones that haven't been bleached. And so as the unbleached ones move into this space, you see the recovery of fluorescence. And by measuring the kinetics of that recovery, you can determine how quickly things are diffusing within the membrane.
The lateral mobility of membrane proteins can be measured in living cells by FRAP, which stands for fluorescence recovery after photobleaching. For this purpose, membrane proteins are often expressed as fusion proteins with the green fluorescent protein GFP and observed with a fluorescence microscope. A selected area of the cell is then bleached with a strong computer-controlled beam of laser light. Those membrane proteins that are not anchored and therefore can diffuse in the plane of the membrane quickly exchange places with their neighbors and fill back in the bleached area. From the rate of this fluorescence recovery, the diffusion constant of the protein can be calculated. Here, GFP is fused to a membrane protein that lies in the membrane network of the endoplasmic reticulum. After bleaching, we observe quick recovery of the fluorescence, showing that the protein is very mobile in the plane of the membrane. The same experiment can be repeated using a protein that is firmly anchored and not free to diffuse. Here, we observe GFP fused to a protein of the internuclear membrane that binds tightly to the meshwork of the nuclear lamina. After photobleaching, no fluorescence recovery can be seen over the same time frame.
Before I sign off, I want to just talk briefly about detergents, which are mentioned in the chapter in a couple of different contexts. So detergents are, you're familiar with them in everyday life. You use them to wash your hair and your dishes. And the most common one that we use in everyday life is SDS or SLS, which are the same thing, sodium dodecyl sulfate. In the lab, we tend to use gentle detergents like Triton X100, which this is an ionic detergent, which has these ions, and Triton is a non-ionic detergent. Detergents are, just like lipids, they are amphipathic molecules. They have a water-loving head group and a water-hating, usually alkyl side chain. And because they have this structure, they don't form membranes, but they can dissolve membranes. And they can also bind to the hydrophobic regions of membrane proteins that are embedded within the membrane. You can use detergents to poke holes in membranes, which we typically do for staining cells. You use a gentle detergent like this to poke holes in the membrane to allow antibodies and other large molecules to go in the cell. And you can also use special detergents to solubilize membrane proteins to help determine their structure or to study them in vitro. And I just downloaded this video because we've all been thinking a lot about soap and hand washing. And I particularly like this video because I think it illustrates both the way that soap works and the way that membranes are organized very nicely. So this is an image that you've probably seen recently or some version of it a zillion times. This is a painting of the coronavirus structure by David Goodsell, who does these amazing watercolors of different cellular structures, and they're all based on real protein structure, and they're all to scale and so forth. And they give a real, I think, vivid sense of the density of macromolecules within cells.
Washing your hands with soap is possibly the simplest way to flush pathogens down the drain. Soap molecules can interact with pathogens, such as coronavirus, leading to their destruction. Each soap molecule has two parts. The head is hydrophilic, which means it likes to interact with water, while the lipid tail is hydrophobic, which means it wants to get away from water to find similar water-avoiding particles. This is the basis of how soap works. When soap molecules encounter lipid particles on our skin, the tails aggregate around them to form spherical structures called micelles, creating a water-free environment. Similar things happen when they encounter enveloped viruses such as coronavirus. Enveloped viruses carry their genome and supporting proteins inside a lipid bilayer membrane, with the proteins necessary for infection embedded in the membrane. In cells and viruses, these lipids are packed neatly into two sheets with the lipid tails facing inward. The lipid ends of the soap molecules are attracted to the lipids in the membrane. They take advantage of the presence of membrane proteins which can perturb the neatly organized bilayer to insert themselves into the viral membrane. If there is only a small amount of soap, the soap molecules only loosen the membrane. But with more soap, they begin to create micelles around membrane lipids. They are also attracted to hydrophobic amino acids in the membrane proteins, extracting them from the membrane. The structural integrity of a virus is essential for infection. So, washing your hands with soap protects you better from infection than washing with only water, and the longer you lather, the higher your chance of destroying the virus.
We're at the end of the hour, but I'll just highlight this other figure from the book that will come back to in talking about other functions of membrane proteins, and particularly as they interact with the cytoskeleton. We're probably not going to have much time to talk about cell-cell adhesion, but that's obviously a major function of membrane proteins. And then this just shows that epithelial cells that are growing in a layer have these tight junctions that between them that actually create domains, the apical and basolateral domains, and some proteins are confined to be in either the apical or the basolateral domain. This forms a barrier to diffusion of proteins and lipids through the membrane. So those are different ways that membrane proteins can have their mobility restricted. And this shows this a little bit closer up. And then this was just talking about how membranes can be reinforced by proteins that are embedded in them and are connected to meshworks within the cell. All right, take care, you guys, and let me know if you have questions. I've been answering questions on Piazza, and we'll keep monitoring that. And hope you're all well, sort of adjusting to current circumstances, hopefully, and take care of yourselves. All right, bye-bye.